Hi, this is Chris Whips. In our last video, we talked about preparing an Agarose gel. In this video, we're going to load, run, and document the gel. All right, so our Agarose gel has set. You can hopefully see it's become more opaque looking or cloudy looking, I guess. So that's set at this point. We're going to move it over into the gel box here and take off the tape and all that. But first, let's get the gel box ready. So I'm just going to swap places here. There goes our gel. We'll come back to that. And here's our gel box. And so what we want to do is put some of the TBE buffer. Remember, we're not using water here. We're using TBE. This is going to allow that electrical current to pass through this buffer. That's the whole principle of separating out these molecules, is the electrical current. All right, so what I like to do here is add, enough buffer that fills the chambers on both sides, and a little bit on that sort of platform where the gel is going to sit here. And now at this point, we can put our gel tray in here. The reason I like to do this is that it's gonna prevent any air from getting caught underneath the tray, between the tray and the gel box. So by adding a bit of that buffer first, we avoid that. All right, so here's our gel. Still leaving the comb in, we're not touching that yet. We'll take the tape off the ends, just carefully, trying to keep it level. We don't want this gel sliding out of here, so we're being careful about that, mindful of that. Obviously the comb is in there, it's gonna prevent it from sliding, but you still wanna be careful. So just gently removing that tape. Now we actually reuse the tape a few times before it loses its stickiness. All right, and now we can put this tray into the gel box. Okay, question, right? Which way do you put it? We could put it in this way, or we could put it in that way. You wanna make sure the comb is on the left side, usually on the left side, but it really ultimately depends on where your positive electrode is going to be. In this case, our positive electrode will be on the right side. I'll show you the lid here. That's our red hookup here. And our DNA is going to migrate that direction. So it's going to migrate across the gel this way. If we loaded the gel like this, the DNA would migrate for a little tiny bit and then just go into the liquid there, fall off the end of the gel. So we definitely don't want that to happen. So just make sure whichever way you're doing this, your DNA is gonna run to the red. Now if we accidentally put this in the other way and loaded the gel and then realized that we made that mistake, you could just hook up the electrodes the opposite way, just so the DNA would migrate whichever way is furthest. All right, so I'm just making sure there's no air bubbles underneath my gel tray. I saw one there, so I'm just carefully lifting it up and setting it back down. There isn't quite enough buffer to cover the gel, and so I'm gonna add a little bit more. The key here is not to add too much. The more buffer we add, the more electrical resistance there will be. So we're trying to avoid that. So I'm just putting a little bit on top, usually enough to just cover the gel. Sometimes on the ends where the sort of meniscus of the gel is, you can see it's sticking up a tiny bit. That's probably okay. Or just add enough that it covers it up. Again, too much buffer, gonna be too much electrical resistance. This thing will start to get hot because of that and potentially will melt your gel. Okay, so now we're ready to pull the comb. The great idea of doing this after we've added the buffer is that what goes into those wells will be buffer. If we pull the comb and then add the buffer, the buffer will fill in those wells to some extent, but if the wells are small enough, air bubbles might get trapped in there, so we don't want that at all. So what we'll do instead is add this buffer like we did, and now when we pull the comb, what goes into the wells? Buffer. There's nothing else. There's no air down where the wells are. We're gonna pull this up, and we'll do it gently. We usually grasp both sides of the comb and just kind of worry it up gently, and there it is. So we have our wells. That's where we're gonna load our samples. I'll show you a close-up of what that looks like. So we've pulled the comb from this gel, and hopefully you're able to see a little reflection from the light above here, but these wells here, and that's where we're going to load our sample. It's helpful to have a background with contrast, so our desktops in here are black, and that allows us to see the wells quite easily. Sometimes we'll use maybe like a red file folder, stick it under the gel box before we even fill it up here. That gives some good contrast. What we want to avoid is a white background. So this sort of lab bench type covering, this paper here, it's a bit old here, but we don't want that under our gel rig. A white background makes these wells very difficult to see. So if you ever encounter that, maybe you didn't realize and you're having trouble seeing the wells, get something dark like black or red against that. All right, so as we get ready to load our samples on the gel, we want to get them out of the fridge. That these samples were in the fridge. We also want to get out a marker that we can run on the gel that gives us known fragment sizes of DNA that we can use to compare our results or our samples to. And so what we use is this quick load 100 base pair ladder from New England Biolabs. We use this one quite a bit. Depending on your anticipated fragment sizes, you might use something different. So having that known ladder, that's really critical. The ladder, you see it's got this purple color to it, the liquid does. That contains loading dye already. Now what is loading dye? It's a dye that we're going to use that we'll be able to visualize when we run the gel. You'll see that as the gel runs. You'll be able to tell how far your sample has migrated. You can't see the DNA, but you can see that dye. Now our PCR reactions here, they also, in this case, already have dye in them. And so there are some products now that come with the dye, so you can set up your PCR and it already contains that dye. So we like that quite a bit, and we use that a lot. It saves a step. In some cases you might run a PCR given the kind of polymerase you're using, or maybe you're running some other kind of sample on an agarose gel like this by electrophoresis, and you need to add the loading dye to it. And so we have a variety of loading dyes. Here's an example here. This orange loading dye. They come at a 6x concentration usually. You could prepare it differently, but that's what they often come at. And so you would add, say, 1 microliter for every 5 microliters in your sample. Now you could add that loading dye directly to these PCR tubes. Again, we wouldn't do that with these ones. They already have dye in them, but if they didn't, we could add loading dye to them, the appropriate amount. Or something we do a lot is we'll take a strip of parafilm, so this is the parafilm side, and we'll put little dots of loading dye. So if we had 8 samples, we put 8 little dots of loading dye. We can then take our sample, pipette up here, 5 microliters, and mix our sample right on the parafilm with our dye. And then we have sample and dye mixed together. The other thing the dye is going to help with is it usually contains something like glycerol that's more dense than water, and when we load that or go to put that in these wells, it'll allow it to sink, so it'll sink down into the wells. So the dye helps to see the migration, and then that something like glycerol or phycol, something similar, will allow that sample to sink into the wells. So let's see how that looks now. All right, so we're ready to load. Hopefully you can see these wells easily enough. And I'm going to start by leaving the first well open. At the end, I'm going to add that ladder that we talked about. So I'll start on the second well, and I'm being careful to not poke through the bottom of the gel. I use my other hand, sort of rest the pipetter on my other hand to stabilize it, and then just dispense slowly. So just being careful that if I see it maybe leaking or I've kind of missed the mark, I can stop. And then try again. Today I have 16 samples to load and I've got extra space here, so potentially if I messed up I could leave a blank. Make sure you note that in your notebook and then continue on. Alright, so I'm going to just continue to load these and just taking your time, making sure you're getting the sample in there. You're not loading the next sample on top of the previous sample. You don't want to do that. So the pipette tip itself, you are going into the well, but not deep down into the well, just sort of at the top of it. It's going to take a little bit of practice to sort of visualize where that is in space. Let's see if I can move this over here a little bit. See this from a bit of a different angle. Something you might ask is, well, how much am I loading into each of these wells? And we actually leave another blank, another space here, and then I'll start with this next set of samples. I got a couple of different things going here. So, but how much to load? Well, it depends on how deep that well is. You could measure the size of the comb and how deep you pour the gel and do some math and figure out the volume like that. Or we just tend to do this by a bit of trial and error. We know these wells will accommodate up to about 10 microliters. I'm just using 5 today because I want to save the rest of this PCR reaction. If these are successful, I'm going to purify them, do something else with them. So you want to make sure you're getting enough in here that you can visualize the outcome, but you're not overfilling the well. In some cases, if you're trying to get as much as possible on a gel, you could use a comb that's bigger. So we have those as well, and you could load, we could load this whole 50 microliters of PCR reaction on this one gel. And usually we're just trying to see, did we get a positive, did we get a negative, did this work or not work? And then, in that case, 5 microliters is plenty. All right, so the last thing we want to do before we get it going here is put some ladder in. Remember our marker that has the known fragment sizes. I'll do this middle lane first. So I left a lane in the middle open. I will load the ladder there. That loading dye is kind of dark purple, so you can see that one a little bit better than our green dye that's in our PCR mix. And I'm going to do the other lane at the front here, work around my camera. That's done. That should be enough to visualize what we have. All right, so our gel's all loaded. We want to do this, as reasonably quickly as possible, and we're going to now set this up to start running. Okay, we're ready to run our gel. It's loaded, and we can put the lid on. Now, the lids on these different rigs are going to be different, but this one slides on. The electrodes are at the back here, and they line up with the contacts on the main gel box. This is a newer one, so it's a little bit snug. The thing to be really careful about is holding down that gel box if you're kind of worrying on that lid, getting it just on there just so. What you don't want is just sort of be pushing the lid and then jar that whole unit forward. All those samples you loaded in those wells, right now they're just kind of sitting there, and by shaking this up a lot, moving it, that's going to cause a problem. They'll come out of there and you'll have to start again. All right, so we're all plugged in. Now we can get the power supply going. There's a switch at the back on this one. We just turn it on. This is really all set up for what we want. We hit the mode button. This is showing us this is going to run at 148 watts at 3 amps for 55 minutes. And I can just hit start. So there it goes. Now, how many watts, how many volts, all these power supplies are going to be different. The key is to know your power supply, and I'll provide we provide some details in the written protocol in terms of what you're aiming for in terms of that power that you're providing to the gel. So this is what we have set up for this system, and that's what works well. The time on this is set to 55 minutes, really just to give it plenty of time to run. I usually check it after about half an hour and make sure it's looking good, and that should be completely fine for a gel like this where we've got lots of room for this, for our samples to migrate. We have the full length of the gel, really. We could probably run this for at least an hour and it would be fine. We get lots of separation and it would be fine. If you have a smaller gel or a different gel rig, different power supply, the first time you're using something, you want to really watch and make sure it's not migrating too fast, so it doesn't migrate off the end of the gel, or so slow that you come back in half an hour and it's barely migrated. You're going to be there a long time. In that case, you'd want to dial up the power. So let me show you a couple other things before we just leave this to its own devices. All right, so our gel is running now, and one of the first things I always look for is on the negative electrode, which would typically be on the left here, is that there are little bubbles forming. If we look close enough, there are little teeny bubbles streaming up. Maybe we can get it from the other side here, maybe able to see them better. Yeah, you can see that quite well here. That means things are working. If you don't see that, there might be, did you hit run on the power supply? Is there a connection problem with the leads here? Are these in good shape? But you really want to check this. I mean, if you don't, you could walk away and come back and nothing has happened in half an hour. You want to make sure this is working, and that's a great way to check that out. We can already see some separation. Get the right angle here. You can see in where the samples that we loaded, that green dye, it's actually a combination of yellow and blue. We can see the yellow already migrating toward the right there. The green is behind it. The dye migrates at different rates depending on what it is. Our ladder had the dark purple loading dye. It's migrating as well, not as fast as the yellow dye. But that's another key, is this working? It's already migrating. And so that's, within the first 5 minutes. So check for those bubbles and wait a few minutes and hope you should see some migration. So we'll leave this be for about half an hour. We'll come back and check it out. One last thing, of course, though, is if you have your ladder and samples, make sure those go back where they belong. If you're keeping them in the fridge, go put them there. Ladder goes back in the fridge. Check the storage conditions on your various reagents, like ladders. Do they go in the fridge, freezer? Just make sure they're going back to the correct place. Okay, our gel is run for about 45 minutes or so. Keeping in mind we only have the one row of combs, so we've got plenty of room here. There's a little condensation that's formed on the lid here. If I look on the side, see a little bit of purple there. It kind of gives me a hint how far that purple dye has migrated. If I use this piece of white paper underneath, you get a little more contrast. You can see the purple there. There's some of the yellow or orange. And that again just gives us a hint. This how far this is migrated. That orange migrates maybe around the same rate as something that's 100 base pairs or so. So that kind of gives us a sense where the front of our migration is. And this has gone about halfway. So for my purposes today, that's enough. So I'm going to stop this. The main thing here is turn off the power supply. I mean, this is set up so if I pull the lid off, these electrodes are going to come away from the unit, so it's not really a hazard, but you want to still be careful. The best thing you can obviously do is just turn this off. So I'm going to tell this to stop. It stopped. I can look, I can see there's no more bubbles forming on the gel in the box here. That's stopped streaming, that's done. So that's off. I'm also going to turn the unit off completely. And there it goes. All right. And let's open this up and go take a picture. Okay, so like before, we're just gonna ease this lid off, kind of worry it back and forth a bit. Now, you know, jarring this gel box, you don't really want to do that, but it's not as much of a concern as at the beginning where those samples were just sort of gently sitting in those wells and we were worried that they'd get upset. Here, they're nice, everything's embedded in the gel. All right, so you want to be careful when you take this out. That gel can easily slip and slide on the tray. So what I do is I usually put one finger down right beside the gel, it's kind of against where the gel is, preventing it from slipping, and then I'll lift the other side. So this isn't gonna go anywhere, my finger's in the way there. I'm keeping my one hand against the gel, and then I can just tilt this and drain off the buffer. Again, if I let go of this, it'll definitely slide. I can see it starting to slide. So I just want to avoid that. Here's our gel, and I'm going to transfer it over to this tray that has some paper towels in it just to soak up any drips. And we'll go take a picture of that. I will mention you have some leftover buffer in this gel rig. What we do is we have a carboy like this with, it's labeled recycled TBE buffer, and we'll use that about 2 or 3 times before just disposing of it, using some new stuff. So it can be reused, and I'll show you that after we take a photo of our gel. Okay, let's take a picture of this gel. Now, any of these documentation systems might be different. This is the one we use in our lab. That might change over time, and this might be different in the teaching lab. But we need to power this on first. There's a switch down here at the bottom along the front of this, so make sure you're turning or initializing whatever machine you're using. And then this has a tray that slides open, and on here is where we place our gel. And I'm actually going to slide it off of the gel tray. I'm being careful, really. I don't want to get any air bubbles trapped underneath it, so I'm making sure the air comes out right at the end. And I try to center this. If there's too much buffer on here, the gel will sort of drift around a little bit. So I'm just sopping up any excess buffer. Again, centering that as best I can. The gel tray, we could keep it on here. The gel tray will transmit the UV light that's going to pass through our gel here. But it does knock down some of the UV, and the gel trays often have a ruler. I don't know if we can see that on here. That'll light up. That could be actually really helpful in some cases. In this case, we just want to see the gel, so this is going to give us the best image possible. So carefully close this. And on this computer here, we will choose this program called Image Lab. Go a little closer here. We have some built-in protocols. This is a medium gel, so we use this gel red medium. Gel red is the dye we used. The first thing we can do is position the gel. That's one of the options here. And it will show us an image of the gel without the UV light. And so I got it pretty close to centered. If I needed to move this around, I can just open the door on the gel box, reach in, kind of move it around, and I'll show you the screen again. I can see my hand. There's a bit of a lag, but by moving this around, you can get it centered. I'm pretty happy with how that looks even without moving it. So let's take the next step here. The next thing we can do on here is click Run Protocol. So this says light source is warming up. By having turned the machine on earlier, this is already getting warm, so it doesn't take very long. And then it snaps an image. So here is the image over here. And this is looking pretty good for what I'm hoping for. I can tinker with the brightness. So sometimes I find it helps to turn the brightness up. I can zoom in on this, just make it bigger. You can see it a little bit better on the video here. So I've got ladder. Those are those sort of bands down these lanes here. And then here are my amplified products of various kinds. Some of these work better, some of these work not as well. These were some samples I was having trouble with to begin with, so I'm glad to see some of them worked fairly well on this attempt. Some of them maybe not so much. All right, so I can save this image. So in this program, what we do is export. So you go to export under File, Export for Publication. And you can save this as a TIFF or a JPEG, again, depending on what you're thinking. If this is for maybe a publication, you probably want to save as a TIFF. JPEG is fine for probably most other things. I'll give it a somewhat informative name. These are some samples I was working on from some parasites. I'll put the date on there. That just helps me keep track of the files and save. And we can also print these images on our little printer over here. But what we found is printing directly from this gel imaging program is not very, it doesn't give us a very good result. So what we do is we go to that saved image and just open it up with our Windows image viewer, and here click print. Make sure it's the right printer. It's the Sony printer. And depends on the system you have. And here it comes. And there is our gel picture. Now this is for my lab book. We also have that digital image. I saved that. I have a big file full of these gel pictures as digital images. So make sure you have both. And then this, we'd cut this out, or cut out the sort of main body of that image and tape that into our notebook. All right, so the gel is, for us, is done. We don't need it anymore. And so what we can do to finish this up is turn this documentation system off. Slide the gel off of here carefully. And we have a garbage bin over here. This can be disposed of in the garbage. Whether or not that can be disposed of in the garbage depends on that dye that you're using in the gel. So if you're using something called lithium bromide, that might need to be disposed of in a different way. You can check with your university to make sure you're disposing of that in the correct manner. All right, so our documentation system here, it has some buffer on it from our gel, and we don't want to leave that there. It'll kind of dry and crystallize. We don't want that for the next person. And so we just use a little bit of distilled water and gently wipe that up. We're not scrubbing at this. We don't want to damage the surface of the glass here on the light. So this is the UV light. The light will shine up from this box up to the camera up above here. That's how we get that image. And so we're just being careful to clean up any of that residual buffer. And once it's done, we can close this up and we're done. All right, so last step will be to clean up. So let's go back to the lab and just do the final cleanup. All right, to finish up, we're going to clean up our mess. So we have our bottle for recycled TBE. It's been cleaned and ready to go. And I'll just pour from our gel box. And it's good to do this in the sink because if some of it sort of spills over the edge, not really a big deal if it's in the sink. Right into our recycled TBE bottle. That's in there, cover it up, close it up, set that aside. And then everything else can just be rinsed off. So I'll rinse that funnel that I just used. Our gel box, just give it a rinse. This does not need to be scrubbed. Had that happen once. Someone scrubbing and actually could damage the electrodes that are in here. There's no need for that. We also don't want to leave the buffer in that gel box. If you're running another gel right after, that's okay, but you wouldn't want to leave that for very long, like overnight or multiple days. That buffer you use can be corrosive to those electrodes as well. For our gel tray, same deal. We're just giving it a nice rinse and into our drying rack here. And that's it. So we'll leave things to dry. Make sure you wash the combs as well. So the comb that we used, I have a couple here from another gel earlier. Just again, gently rinse them off and then leave them to dry and be ready for next time. All right, so that's how you prepare and run an Agarose gel.